Okay, alrighty. Let's get started. Thank you all for coming today. Especially given since next week is Reading Week. Yay! Woohoo! Reading Week! It's a chance for us to relax, recuperate, catch up with any of the stuff that we need to catch up on. So awesome. That's why I'm particularly happy to see you guys here today, because then you have a whole week off. Today we have a very exciting lecture that really, I'm going to describe to you the story of a woman who really went against the fixed dogma of her time. So it's really exciting to tell you her story. That's of Barbara McClintock. And that's, her revolutionary idea that you could have genetic elements that are not residing in fixed places that can somehow move from one place to another. And I can't, describe to you how incredibly unusual this idea was at the time. I mean, this was even before the structure of DNA was known, before the fact that, really almost concurrent with the idea that it was even DNA that was the hereditary unit, right? She was operating way before then. So, and then, of course, it turns out, as you all know, that these things that she discovered, these transposable elements, they turn out to be everywhere. They are so ubiquitous. And it's something that, when it was first proposed, she was not believed at all. Okay, now before I start with my lecture, though, I've been promising, I did want to give you an idea of the flavor of some of the types of questions that you might see on the final exam. I have put up, there's a example final so that you can see the types of questions. As I said, this section is not, it's not, certainly not as focused on problem solving as the previous section of the course was, but there are a few problem solving questions. I really think that they're not very difficult. This would be an example of the types of problem solving questions that you might find on the final exam for for these types of, for this section of the course. Okay, so I want you to read it, and we're going to, I like to have full participation, so I don't use clickers, but I would like you to raise your hand after you have thought about this, and don't be shy. No one's recording your answers. You just get to raise your hand after you think about which answer might be the right answer, okay? And don't worry if you're wrong. I give points for originality, too. Okay, so I'm going to let you read this and think about which answer you're going to vote for on this nice Friday right before Reading Week. And remember, you don't have to vote the same as your neighbors. There's nothing at stake here. Nothing at all. Have you guys all read the question? Yep, okay. Are you ready to vote? Voting with your hand, remember? Voting with your raised hand. Okay. So, I'm going to start from the bottom. Everybody's ready to vote, right? Everybody is ready? Okay. Who votes for D? D. Okay. Who votes for C? Okay. B? Okay, some hits, some hits. Who votes for A? All right. All right. Good Good for you guys who bucked the trend, but it is A. All right, so here is what the solution is. The solution is you're thinking about, okay, what kind of cross could be happening? You got some clues that this might be a quantitative trait or not. Remember when we talked about quantitative traits, one of the key things was additive effects across loci, okay? So in this case, there would be two loci with additive effects, where the additive effects are, in this case, number of hairs, okay? With each dominant allele contributing 10 hairs. You would come out with the appropriate variation where if you did not have any of the dominant alleles, you would have 0 hairs. If you had all of the dominant alleles, 4 of them, then you'd have 40 hairs. So you would see this distribution of phenotypes that could range from 0, 10, 20, or 30 hairs, okay? And so that means that the answer is A. Very good. You guys are on the ball today. I'm very happy about that. Yes, give yourselves a hand. Yay. Okay. All right. Excellent. Yes. Okay. All right. So today's lecture is about transposable elements in maize, because that was Barbara McClintock's model system of choice. It's a very inspiring story about persistence and belief in the process of scientific discovery that was not vindicated for decades after she made the original discovery, okay? But finally, her discoveries were vindicated, and it turns out that transposable elements have turned out to be not just very ubiquitous, but also incredibly valuable tools for research, genetic mapping, etc. Your book has a nice series of portion of a chapter on this. I encourage you to have a look. All right, so for the story of Barbara McClintock, she discovered what she called these mobile elements in the late 1940s, but it was not until 1983 that her discoveries were actually widely accepted and she was awarded the Nobel Prize. So let me tell you about her story. She used maize as her model system. Maize is a member of the grass family, right? It's a member of the grass family. It was domesticated thousands of years ago by the indigenous peoples of Central America from the wild species called teosinte, right? And it has, eventually it became domesticated and is known as corn. All right, now it turns out, and part of the reason why she used maize as her model system is because, number one, they're easily discerned and very interesting phenotypes that they have. For example, here, right? Where you have things like color and shape that are easily viewed and screened in breeding studies. But number two, because remember, this was in the 1940s, before we have modern molecular technologies available. It turns out that fortuitously, maize chromosomes are very large and they are easily visualized by light microscopy, okay? So she happened to notice that in one strain of maize, chromosome number 9, one of the chromosomes that were identified by karyotyping, seemed to break very frequently at a particular site, all right? And so based on her very careful observations, she was able to propose that this might be due to a new type of genetic element, right, that can move around in the genome. And as I said, this was like mind-blowing at the time. Nobody thought this was possible in their wildest dreams. All right, so her work on the cytogenetics of maize using light microscopy allowed her to develop this theory. And she called these genes controlling elements that could move around, right? So she hypothesized that this one strain of corn of maize that had frequent breaks in chromosome 9, she hypothesized that the breakage was due to two genetic factors: a DS or dissociation factor located at the site of the break, right, as pictured here, plus another factor that she called an activator, an AC element, right? And she proposed that that was an unlinked genetic factor that was necessary to activate the breakage at the DS locus. She suspected what she called mobile elements when she found it impossible to map the AC to a consistent location, right? She found that it seemed to jump around. All right, so, and as I mentioned, this was a very groundbreaking idea. Now it's completely accepted, but at the time, nobody thought that this was possible. So even though she published her work in the early 1950s, it was not widely accepted for decades until the 70s and 80s. And now, of course, it's been, confirmed using modern molecular techniques, all of the details by which this happens, all right? So, it turns out that part of chromosome 9, right, as pictured here, controls several phenotypes of corn kernels, as depicted here, right? These different loci control things like pigment, the C locus. You can have alleles that are able to produce pigment versus colorless ones. You can have another locus which controls how plump or shrunken it is, the shrunken locus. You can have another locus that controls how shiny it is, the waxy locus, shiny versus not shiny. And they're all located on this end of chromosome number 9, all right? So what can happen is that if you have chromosomal breakage at the DS locus, as you can see here, which is located closer to the centromere than all of these other loci, if you have chromosomal breakage at the DS locus of a heterozygote, it could result in loss of the wild-type alleles, right, here, revealing what is normally hidden in terms of these recessive alleles, right? And then the recessive alleles, a break can occur in a single cell, which then divides mitotically to give rise to a whole section of the kernel. That's why this scenario here turns out looking like this. All of these are wild-type cells in which there is no breakage, and so it's heterozygous, and you see the dominant phenotype, but in all of the mitotic descendants of a single cell in which this breakage occurred, you will see the recessive phenotype uncovered, okay? And then you can also have a scenario in which you have insertion of DS into the C gene, right? The gene controlling pigmentation. So if you have this scenario, then what you have is colorless, the colorless kernels, unless in certain cells the DS jumps out, right? It's activated by the presence of an AC element, allowing it to jump out in certain cells and all of their mitotic descendants. Because when it jumps out, then all of a sudden you have a reconstitution of the dominant C allele, which allows for pigmentation. So that's why in these instances, each time you have a jumping out of the DS element, you'll have a spot, right? So all of these are independent events of jumping out of the DS element and reconstitution of the C allele and all of its mitotic descendants. That gives rise to these spots. So this type of unstable, what we call unstable phenotype, that tends to revert back to wild type, is a clue to the participation of mobile elements, all right? Okay. So this basically summarizes what Barbara McClintock surmised from her breeding studies of these maize strains. She surmised that control of the way that the kernel pigment looks resided on a particular locus on chromosome 9, where the normal C gene, right, the normal dominant allele of the C gene, gives rise to a pigmented phenotype that looks like that, right? But if you have insertion of this DS element into the C gene, it disrupts it, and you get a colorless phenotype, all right? And this colorless phenotype is stable if there is no AC present. So it's completely colorless. There are no spots, right? However, if you have a AC element present somewhere else in the genome, doesn't have to be right next to it, right? Could be totally unlinked. It can catalyze the DS element to excise itself, to remove itself from the C gene, and you get reversion back to the wild type in those cells. So you get these spotted kernels, as I explained in the previous slide. And then she also discovered occasionally that some strains seem to have an AC element in the middle of the C gene, right? And that this situation is always unstable. It always leads to the spotted phenotype and not the colorless phenotype, right? Because the AC is capable of mobilizing itself. It's not like the DS element. So, and then she also found that occasionally an allele of the AC type could actually transform itself once it had jumped into an allele of the DS type once it had inserted. But that was pretty rare. This implies that DS is probably an incomplete version of the AC element, okay? So, definitions: autonomous elements like AC, they require no other elements for their mobility, whereas non-autonomous elements like DS, they require other elements for their mobility. And just bear in mind, she was able to deduce all of this from light microscopy combined with breeding experiments, all right? It took almost 50 years after her initial studies to actually identify the molecular underpinnings responsible for these unstable mutants. So they're the experiments that showed actually what was responsible. The AC and DS elements were actually isolated, sequenced, and they were shown to be related to DNA transposons, such as the P elements of Drosophila, which I'm going to talk to you about in a minute. So now we know a lot about the molecular mechanisms underpinning all of those studies that she did originally using light microscopy in maize, all right? So what's known now is that the AC elements have these terminal inverted repeats, as you can see here. It's got a protein coding gene that codes for a single protein, a transposase, right? The AC element. The DS element, on the other hand, does not encode for a functional transposase, and so therefore that's why it can't transpose on its own. The transposase enzyme that's encoded by the AC element, as depicted here, can bind to the ends, those terminal inverted repeats of either the AC element or the DS element, and promote their transposition and reintegration into a new site, okay? So that's why some of the molecular mechanisms underlying these transposition events. Let's look at it in a little bit more detail. So details of the insertion into a new location. The transposase actually makes a staggered cut, as you can see here, very similar in the target site DNA. And that staggered cut is very similar to restriction enzyme cuts, right? So the transposase makes these staggered cuts, And then the transposable element inserts between these staggered ends, and the host repair machinery fixes the ends using the complementary strand. So that's why you actually, in the end, this results in repeat elements on either side, right, of the transposable element, which are direct repeats. So they flank the mobile element at the sites of the insertion. So it's a target site duplication. Virtually all transposons do this. They're flanked by target site duplications. So that implies that, in fact, very similar mechanisms are used across different types of transposons for insertion. Now, remember that I said transposable elements also tend to have inverted repeats at their ends for recognition by the transposase. That's different from the target site duplication, which is a direct repeat. Okay. As I said, transposable elements, it turns out, contrary to the beliefs of many people, they turn out to be incredibly common, right? Some of them in bacteria, some of the most important bacterial transposable elements are contained on R factors, plasmids containing genes that encode resistance to antibiotics. These factors are transferred during cell conjugation. And many of the drug resistance genes known to occur on R factors and other places, in fact, reside on transposons. So transposons are absolutely critical in helping to move important pieces of DNA around. And then even that's prokaryotes. And then in eukaryotes, the situation is even more extreme. You know, one of the really big surprises arising from all of the genome sequencing that was done of eukaryotes was how much of the genome is actually comprised of transposable elements and other types of repetitive sequences. You know, up to 1/3 of the human genome is composed of transposable elements, which seems amazing. Now, eukaryotes have two different main categories of transposable elements: the retrotransposons and the DNA transposons, and I'm going to talk about each one of them in a little bit more detail. All right, retrotransposons. Retrotransposons are actually quite similar to retroviruses in that they both have an RNA intermediate. So in this case, there's an RNA intermediate that then gets reverse transcribed back into DNA and then reinserted, okay? So that's one of the major differences between this type of transposon and other types of DNA transposons. This type of retrotransposon is actually quite highly variable. Some of them have LTRs, or long terminal repeat sequences, that can be several hundred bases long. So this type of transposon can actually be quite large. And some of them even have things like poly-A tails. Retroviruses are, in fact, more similar to the bigger types of retrotransposons, the LTR retrotransposons. Both retrotransposons and retroviruses tend to have these genes. First of all, they have to have the Pol gene, which encodes for a reverse transcriptase endonuclease, right? Because that is what allows for reverse transcription and insertion back into the genome. Some retrotransposons also have the GAG gene, which is a protein with a role in RNA maturation. Some of them have poly-A tails, as I said, and others do not. One of the big differences with retroviruses is that retroviruses have to have an additional gene, the ENV gene, which is, involved in encoding the structural protein surrounding the virus and is what allows it to infect other cells. Retrotransposons, generally speaking, do not have this gene, although actually there have been some rare ones that do. But retroviruses have to have that. So the big differences between retrotransposons and retroviruses is that retrotransposons do not infect other cells. They can only jump around in the genome, in the cell in which they occur, all right? So, and they encode reverse transcriptases, which mediate this reverse transcription back into double-stranded DNA and then insertion into a new location in the host genome. All right, so this is in contrast to DNA transposons. And so this is just a diagram of the mechanisms of a certain type of DNA transposon called a class II element. This is one of the simplest types of DNA transposons. This was one of the first transposons that was discovered. For example, the DNA transposon that was discovered in maize by Barbara McClintock. It is also the first type of DNA transposon to be characterized at the molecular level, and that was the P element in Drosophila. So this is a description of how P elements work. Now, P elements really resemble the simple transposons of bacteria. Its ends are short inverted repeats. They tend to be very small. It only encodes one gene, which is the transposase required for mobilization, right? The transposase enzyme, as I mentioned, recognizes the inverted repeats and is able to excise the P element. So here it's being excised. It can then be inserted into a new target site, right? A new target site in the genome. And when the P element integrates into this new location, as catalyzed by the transposase, it, as I mentioned, creates a duplicate of the 8 bases present at the new target site. So as you can see here, these direct repeats, right? New target site duplication. However, you might ask, what happens at the old location where there used to be a P element, but it was excised and moved to a new location? In fact, what happens at the old location is that there's a gap. That gap is increased by the action of exonucleases, right? And then the processes of DNA repair happen. And depending on whether the repair is made using a sister chromatid or homologous chromosome containing a P element or not will determine whether or not the P element will appear to remain in the original location or be removed. So this is a case in which a sister chromatid or homologous chromosome used for the repair contained a P element. So then it's repaired to contain a P element in the original location, okay? And this is a case in which the homologous chromosome was lacking a P element, and so it was repaired and there's no P element there. So depending on how the processes of repair happen, then you will either retain the original transposable element or not, okay? Now, in humans, there are several types of transposable elements that are very common. And, large genomes like the human genome tend to be like this. And that has what's contributed to something called the C-value paradox, where, in fact, the size of the genome doesn't seem to correlate very well with biological complexity. For example, salamanders have genomes that are 20 times the size of humans, right? Why is that when they don't have 20 times the number of genes? In fact, a large proportion of genomes in multicellular organisms and eukaryotes are composed of repetitive sequences, many of whom are related to transposons and transposable elements. So the vast majority of transposable elements in humans come from 2 different classes: the lines or long interspersed elements and the signs, short interspersed elements. Lines are actually very similar to retrotransposons, as you might have guessed. They tend to encode things like reverse transcriptases, which they use to move around the genome, right? Signs, on the other hand, are more like non-autonomous lines. They're similar, but they don't tend to encode. They tend to be much shorter. They tend to have lost their ability to move around, to mobilize by themselves, and don't tend to encode their own reverse transcriptase. Some of the most abundant sign elements are actually ALU elements, that contains, they're named ALU because they contain target sites for the ALU restriction enzyme. And the human genome is known to contain over 1 million ALU elements, or 10% of the entire human genome. Okay. You might ask, and this is a very valid question, how do organisms survive with so many mobile elements jumping around their genome? They can jump in and out anywhere. And as you just saw, if they jump into a protein coding gene or some part of the genome that's important, they tend to disrupt function, right? So how is it that we can exist with such a huge proportion of transposable elements in our genome? And the answer is that, in fact, a huge proportion of the genome is non-coding. Often, the insertion of mobile elements is into parts of the DNA that is not as important. They're often inserted into things like introns, right? And even, and number two, they're often found to be defective. So they may have jumped in, but then they're defective, and then they're basically immobilized and unable to transpose again due to mutations. Lack of inverted repeats, lack of an active transposase. Another mechanism to protect against the mobilization of transposable elements are host regulatory mechanisms such as epigenetic changes. So, in other words, safe havens if they jump into things like heterochromatin. But despite all of these things, of course, sometimes mutations do occur that are attributable to the actions of mobilized transposable elements. So some famous examples are, for example, the line transposable elements that disrupted the factor VIII gene, causing hemophilia A. Another really famous example is the ALU insertion into the BRCA2 gene, which is known to be associated with breast cancer. Okay? Now, there are many ways you can imagine that transposable elements would mess things up. This is just one example. Some of them are very long and can actually promote unequal crossing over by mispairing, right, during the normal process of recombination. Because the transposable elements themselves can pair, they can promote things like duplication of segments of the chromosome and duplication and deletion of entire genes. All right? There can also transposition or mobilization of transposable elements can also be involved in relocating genes to other parts of the genome that they're not supposed to be. And one of the ways that this can occur is because of the mechanisms by which transposable elements excise themselves and relocate to a different part of the genome. And the fact that they use these inverted repeats, right, mean that if you have two insertions of two different instances of transposable elements that are close enough together in the genome, okay, so this is two separate insertion events, right? Because of the mechanisms by which transposases recognize inverted repeats, what can happen is that instead of the transposase that comes along that's going to mobilize the transposable element, instead of recognizing this inverted repeat and this inverted repeat, or recognize this inverter repeat and this inverter repeat. Instead, it recognizes this one, and then excises this entire region and moves it to a new location. And so you can see that actually, depending on the particular transposable element, as much as 400 kb of genome can be moved this way to an entirely different region of the genome, right? Because it can be moved anywhere that the target site duplication, the target site recognition sequence occurs. So that means that all these genes in between here, like for example this white gene, are moved to an entirely different chromosomal location, could even be on a different chromosome. And what happens when you move genes into a completely different context is that all of the regulatory mechanisms are changed. The whole genetic background in which the gene is operating is changed, which means that it's likely to be expressed at a time and place, and maybe even in an amount that's inappropriate, okay, and going to cause problems. All right, so this issue with the level of expression of the gene, exactly what tissues it's expressed in, exactly when and where it's expressed, usually will change when this happens. Okay. And then finally, transposable elements have been enormously useful in laboratory research, and none more so than the p-element mediated transposable element in Drosophila that has been used to great effect to to easily and efficiently insert genes into Drosophila. And this is just a description of how that process occurs, okay? So you have one vector here, right, which is basically a deleted p-element. It's a p-element with the gene deleted, and instead of the transposase gene, you're putting in a gene of interest. In this case, we're putting in the rosy gene, which is a marker for eye color. It's engineered into a deleted p-element, right, that is carried on this vector. Okay. Simultaneously, along with that, you also inject a helper plasmid that contains an intact p-element. Both of these are injected into an embryo that is RY minus at the rosy locus. Okay? All right. So then after the injection, your wild-type rosy gene mobilizes itself with the help of the intact p-element into the chromosomes of the germline cells in this embryo. So the embryo is rosy minus, right, homozygous, but in some of the germ cells, this rosy locus has been inserted, right? So you grow it up to the adult. The adult is rosy minus in terms of its phenotype. But some of its germline cells, right, in which the p-elements were mobilized, have rosy plus alleles, right? Note that this p-element, this is not a homologous recombination. The p-element has inserted this gene randomly somewhere in the genome, according to how transposons always mobilize, right? Somewhere in the genome, but in this case, it's been engineered to occur in germline cells, is what you're going to see. And these cells, when you cross with rosy minus, some of the progeny in which the rosy wild-type gene has inserted into the germline cells, some of the progeny will have rosy wild-type phenotype in terms of the red color of their eyes. All right? So it's a way to be able to easily make transgenic flies. You could imagine, in addition to the rosy gene, you have a gene of interest, right, that you're trying to insert into the genome that you could put along with the rosy gene. And then you could use the rosy gene as a marker to screen for where you have successfully inserted your gene of interest. All right? So it allows for the transfer of any gene of interest to quickly and efficiently make transgenic flies. Okay, thank you all. Have a great reading week. Enjoy yourselves. I'll see you the week after that.